Yours. At least we have number 2. All right. Usually I don't need a microphone anyway, but I think I'll try and speak into it because of the recording that's going on here. All right, so welcome everyone and people still coming in. This session is about the world around us in this building, basically, and other indoor environments. So this session was convened by Brendan Bohannon, who works on a lot of related topics to what this session is about. Brendan is not able to be here, and he, unfortunately for you, asked me to be his replacement. So I'm Jonathan Eisen. I'm from UC Davis, and the session, as I assume most of you know, but just in case, it's like Southwest Airlines or something if you're in the wrong place. Session is on the great indoors, recent advances in the ecology, or really microbial ecology of built environments. And if you haven't been posting to Twitter and you want to, that's the hashtag that people are using for posting to Twitter related to this meeting. If you aren't familiar with Twitter and you want to see what's going on at the rest of the meeting, you might want to check out, just log on to Twitter and search for that hashtag. There's about 30 or 40 people around the meeting posting about various talks and other things going on, at least. So what we're going to do today is go through 7 talks in this session. I'm going to give the first talk in replacement for Brendan. Again, apologies if you were coming to see Brendan. I didn't dress up quite as nicely as Brendan normally does, but this is pretty nice for me. So then second talk will be Nicholas B. on, I sort of just gave the headlines for their talks here on the air microbiome, and then Katie Kirsch on hotel room microbial studies, Mark Hernandez, another talk on airborne microbes, although obviously different from the first one, John Senko on flue gas associated microbes, Kimberly Ross on drinking water microbes, and then the cleanup hitter here, not the pitcher batting ninth, but the cleanup hitter here is Jordan talking about aspergillus and a variety of things related to aspergillus. So what I'm going to do for the first talk here is give you a little bit of introduction to microbiology of the built environment, not a lot, but a little bit, and also talk about this project that I'm running called MicrobialNet, which is the microbiology of the built environment network. And I'm going to give you a little bit of history behind this project, and also some of the things that we're doing associated with the project. And please, if there are any questions along the way, just raise your hand, shout them out. I'd like to have this be a conversation. If anything comes up and you want to ask a question about it, please do so. So I assume many people here have already thought about these issues, but why should we care about microbes in the built environment? I didn't really think about microbes in the built environment until a few years ago, and after I started getting involved in this project, this is the sort of #1 topic that comes up if you say you're working on microbes in buildings or you're trying to convince some funding agency that is of interest. They ask, first of all, what is the built environment? Which I'm still not 100% sure exactly how I would define it, but I will talk about that. And then why should you care? So to me, the built environment is really any place you would really call sort of indoors or an internal environment that is shaped by design and construction by people. So buildings, planes, cars, etc. Humans shape our entire landscape. So some people view roads as the built environment. I'm much more focused on the built environment as places that are sort of enclosed. And I don't know if that's sort of the standard definition, but that's what I focus on in terms of the built environment. And there are many reasons to be interested in this built environment. One of them, which people always sort of describe and cite in their papers, is that we spend a lot of time in the built environment, and therefore we should be concerned with what the other organisms are doing in our environment. So it is really our niche, or a large part of our niche, and obviously some people spend less time in the built environment and other people spend more time in the built environment. but it is a large component of the niche of people and many plants and animals, actually. And actually, the reason I'm personally interested in the built environment is really I view it as a model system for studying microbial ecology and microbial diversity. So unlike, say, some external environments, which unfortunately are hard to control, although the built environment isn't perfectly controllable, we can define the built environment in many ways. We can construct facilities with known components and with known processes going on in those built environments. We can somewhat control the inputs and outputs in that environment, probably more easily than we can, say, in the surface ocean waters or some soil environment. And although you can do this in the external environment, in theory, especially with like tract housing and other things, we can make many replicates of the built environment where the environment that you're studying is very similar across different actual sites. And again, of course, the microbes in the built environment likely play a role in things that people care about, such as human health directly in some ways, say the pathogens that are in the built environment. The microbes in the built environment probably do something to shape the human microbiome, so the microbes that are in and on us, on our skin, in our gut, in our mouths, we are sampling from our environment in part. Some of our microbiome comes from our mother and comes from our interactions with other organisms, but a lot of it is going to be influenced by the built environment and vice versa. The human microbiome is shaping the built environment, at least in some part. And of course, there's also sort of, even if you don't consider the microbiology of the built environment, there are many people interested in what you could call the ecology of the built environment. And the microbes play a role in that ecology in shaping the particulate matters, in shaping the volatile organic chemicals, and shaping a lot of the built environment. And interest in microbes per se in the built environment is nothing particularly new. People have, of course, always been interested in, say, how we are going to transmit a pathogen inside an airplane, since we've had airplanes. And someone was telling me that on the flight over here, they were saying this in the press conference earlier, on the flight over here, they showed Contagion on the plane. Not really the smartest selection. But people have been concerned, of course, with transmission of pathogens, whether they come from surfaces or come from the air or come from direct contact. And most of the focus on microbiology of the built environment historically, like many other areas of microbiology, is focused on the pathogens, certainly for human health-associated studies. And most of the work has been done using traditional culture-driven methods. And what a lot of this session, a lot of the talks in this session are about, and a lot of the sort of buzz that I will be talking about in microbiology of the built environment relates to, is that there have been technological changes that have happened in the last 5, 10, 15 years that allow for new approaches to be added on top of the traditional approaches in studies of microbiology of the built environment. And one of those, which I work on and which I will talk a little bit more about, is DNA-based studies to do culture-independent studies of the microbes in the built environment. And I assume most people are familiar with these types of studies, but DNA-based studies, originally with ribosomal RNA PCR and now with things like metagenomics, have been applied to many microbial-dominated ecosystems, human microbiome, oceans, etc. And that because DNA sequencing costs and difficulty have plummeted over the last few years, the ability to use DNA as a strategy to sample the community has become much more readily available to many people. And so this also, of course, applies to culture-based studies. So we can now sequence the genomes of everything that someone has cultured. We can apply other omic-based technologies and other high-throughput technologies like proteomics, metabolomics, et cetera, which can allow us to characterize all of the molecular components of the microbes either in an ecosystem or in culture. Doesn't necessarily tell us everything that they're doing, but these techniques, because they have become more readily available and cheaper, make things sort of possible to add to the current arena of work. So, but, there's always a but. So culture-independent studies, which I'm very interested in, have taken off in the human microbiome. For example, there's a session on Tuesday here at the meeting reporting on many of these new human microbiome papers. There's been big projects in Europe and in the United States. Prior to that, there were lots of people applying culture-independent methods to the oceans or acid mine drainages or other ecosystems of interest. And in the last 10 or 15 years, that has taken off in other ecosystems, but has not quite taken off in studies of microbiology, the built environment, until very recently. And there are reasons why one area might not adopt some of these new approaches as rapidly as others. And the same is true for genomics. So genomic studies of microbes in the built environment maybe perhaps have lagged a little bit behind. So one question you might ask is, how do you accelerate progress? Why is it that there's this gap in application of these new culture-independent methods and high-throughput methods to microbiology, the built environment? And you can identify lots of things that you might be able to use to help fill in this gap, whether it's obviously money is an important thing, just interest of people, getting the right people involved to work in the area, openness with access to samples and access to technology, communication about that information, education, etc. And I mean, clearly, money, if you don't have money to work on it, that's not really going to happen. And the reason I got really drawn into this area, partly because of money, but partly because of the people that were working on it, is that the Sloan Foundation, the Alfred P. Sloan Foundation, got interested in studies of microbiology, the built environment, in the last few years. Originally, this was probably about 7, 8 years ago or so. They funded a couple of pilot projects in areas related to molecular studies of microbes in the built environment, partly because they were interested in biodefense and in sampling buildings, such as sampling the air in buildings. And if you go into a building and you sample the air and you want to apply molecular methods to study the microbes in the air, and you don't know what's in the background, what's normally supposed to be there, how do you flag something as a potential biological attack if you don't know what's supposed to be there? So I think that sort of spurred the Sloan Foundation to say, wow, we really don't know a lot about the ecology of the microbes in the built environment. And they booted up a new program that Paula Olszewski is the program officer of. I don't know if Paula's here or not. But they basically announced a few years ago a program in this area. And they focus on things that are sort of gap funding type programs. So they announced this program. They put out calls for proposals in 5 different areas. One of them was for a database type project. One of them was for a center related to microbiology of the built environment. And one of them was related to a communication network related to microbiology of the built environment. And after some pestering, they convinced me to apply for this. And that's what I'm going to tell you about, is this project that we've gotten that we call MicrobialNet, which is the Microbiology of the Built Environment Network. And this is a collaboration between UC Davis and a few people in my lab at UC Davis, and Hal Levin, who's at the Building Ecology Research Group and is right over there. And a few other collaborators have been involved, and many of the people funded by the Sloan Program have also been very helpful in this project. And what MicrobialNet is really tasked with doing, you can view this as an experiment. This is not a common thing for a funding program to initiate right when they start up a program in a new area. And what we're charged with doing is really working on these 3 tiers. So we're trying to do network-type activities that I'll tell you about in a minute related to these 3 groups. Those funded by the Sloan Program, so people who are getting money as part of this project, which is a small number of people. There are about 20 grants that have been awarded. And obviously, if you want to reach out to people outside of that, you can't focus on only the people that are getting funding. So we're also working on multiple things related to people in the broad area related to, that could work on something related to microbiology of the built environment, whether they're building scientists that are surveying the air in buildings but haven't looked at either microbes or molecular studies of microbes, or they're microbial ecologists that study the human microbiome and are interested in how hospital-acquired infections get transmitted, etc. we're trying to sort of grease the path to get those people interested in working on microbiology of the built environment. And then Sloan has this term they like to use called the stakeholders. That's basically sort of people who might control funding related to expanding this as a program. We also view the public and public outreach as an important component of this. So what we're doing, and again, I view this as, I spend a lot of my life in scientific communication and scientific social media, all of which prior to this project was unfunded. So this is new to me to actually get funding to work on things related to this, and we're sort of playing around with a lot of different areas, and we're basically doing these 5 activities related to this project. We have a website /hub that I will show you in a minute. That's microbe.net. A website or hub is useless if it doesn't have content. So the fact that we have a website, who cares? But that's where the portal is for some of the information that I'll be telling you about. So one of the main activities that we're actually doing, and I would love input from any of the people here on this, is we're trying to identify areas within this field of microbiology of the built environment that could benefit from workshops, meetings, conferences, etc. So we have some funding to our grant that now we are able to use to fund, say, a small workshop on a particular topic, and I'll show you some of these that we've done already. But we're also interested in if people flag something, say there's a panel at a meeting that says, oh my gosh, if we only could bring together these 6 types of people, and that will revolutionize studies of microbiology of the built environment, we can actually work with you to fund that type of meeting or gathering. We are creating and curating resources, and I'll give you examples of these. We're experimenting with scientific social media. I will tell you about some of these. And the last thing that I'll tell you about is really the ultimate goal, which is to get everybody else doing all of the things we're doing, rather than just sort of having to catalyze these activities ourselves. So like I said, there's the website, but again, website is sort of useless if it doesn't actually do anything. So one of the things we've been doing related to meetings and workshops is identifying sort of large conferences that are on a particular topic, say microbiology like ASM, or indoor air meeting, and trying to organize interactive, interdisciplinary gatherings at those meetings that try and bring together people from the building science side of things with microbiologists, the traditional kind, if you want to call it, as a separate area, with molecular, culture-independent microbiologists, and try and get them to work together and spawn off collaborative projects where people are going to, apply the same approaches that have been used in oceans and the human microbiome, et cetera, to microbiology of the built environment. We've done a few of these at Indoor Air, at AAAS. I could go through and give examples of all of these. One that I will tell you about, which came up just sort of as a side project, we organized a workshop at UC Davis on citizen microbiology, because we think that getting citizens, people involved in thinking about the microbes in the built environment, would be a really useful way to both get people thinking about microbial diversity, but also to get data from people helping collect samples. And there are already now multiple examples of people doing this. Jack Gilbert in his lab at Argonne is doing a home microbiome project, Rob Dunn, and others. I think Jordan and a few people are doing another sort of home biology project, which includes surveys of microbes, where people are sort of helping swab or collect samples, and then they send them off, and you do some molecular study, and you're trying to engage them in the same way that, say, the Cornell bird observatory projects engage people in studies of birds. Can we do that with microbes? How can we do that with microbes? So again, like I said, we would really love ideas in this area related to workshops. For resources, we've been trying to identify sort of topics or tools that would be of benefit for bringing people together in this area. So one thing that has been flagged at every meeting that we've organized is that the jargon that is used in microbiology and in molecular surveys of microbes is very unfamiliar to many of the people on the building science side of things, and the jargon used in the building science side of things is very unfamiliar to many of the microbiologists. So we've been trying to write and produce simple fool's guides types of things for our website, which we have done for things like what is a molecular survey of the environment, what are microbes, what are the different kinds of microbes, anything that we think can be used by people who would like to approach this area, but maybe the reason they're not approaching it is some terminology issue or some background issue. We would like to produce those types of documents. So we have a page on all of these simple guides that anybody can get access to. And by the way, all of the material we produce is done with a Creative Commons license, so anybody can download any of this material, use it in any way you want, reproduce it, modify it, etc., to try and spread the information as broadly as possible. We've flagged a few of these areas. We have a list of about 10 more, but again, if people are interested in microbiology, the built environment, and you think there's some area that would be useful to have a background guide, we can work to produce it. So if you just sort of let us know. One area that we think is really, really important and is a new growing area of scientific social media is sharing references and sharing information about new publications in a social environment. So everyone's overwhelmed with the literature that is out there. Keeping up with the scientific literature is overburdening and almost impossible in many fields. And one way to help keep up with that information is to build a social network around rating, ranking, commenting, etc. on scientific publications. And there are a few examples of this out there. We've ended up working primarily with a system called Mendeley, which is really completely brilliant, sort of socially aware reference collection. So you can imagine it as EndNote, but mixed with Facebook. And what it allows you to do is, we have a group on microbiology of the built environment, for example, where there are about 50 members in this group. And if one person finds a paper of interest, they post it to this group, and then all other members in the group can get a little flag saying so-and-so posted this paper. They can post comments on the paper. If you wrote the paper, you can actually post the PDF to the group, and then everybody in the Mendeley ecosystem has access immediately to the PDF of your paper. It's sort of like an author's website. And this type of sort of social referencing, social discussion of the literature is a great way to get people to interact on some newly developing area. And we've used this in many other projects. It's a little bit harder in this area because people span such wide topics, you know, architects and building scientists and air conditioning repair people and microbial ecologists and computational biologists. Getting them all to play in the same sort of social network and sharing information is not necessarily easy, but we think that these methods are going to be very useful at getting people to keep up with the field and basically added value by working with other people. We have information for those interested about the entire Sloan program, so we were sort of more nimble at creating web materials than the Sloan Foundation was, so they asked us if we could just post everything to our website rather than them having to go through the bureaucracy of posting new things. So we have links to all of the currently funded Sloan projects that work on microbiology of the built environment. We have some detail about the Sloan program itself, and we post, when they have calls for proposals, as they recently did, we post information about those calls for proposals. We have a variety of other resources. We're posting meeting reports for all the meetings we organize, as well as other meetings that we go to. We've got compilations of government reports that relate to, say, toxic mold, or microbiology of the built environment, or molecular surveys of microbes, funding opportunities, etc. And again, we would love anybody who works on things related to microbiology of the built environment, if you flag things as being of interest, if you have resources that you want to share broadly, tell us about them and we can add them to our collection. And you know, as I mentioned with Mendeley, but also we're trying with other areas, we really do believe that scientists should use social media for more than just sharing information about their personal lives. that social media can be very powerful in sharing scientific information as well. And so Mendeley is an example of this, but Facebook groups are used by many large organizations as a way of building community and sharing information about upcoming meetings, about new publications, etc. We have a blog through the MicrobialNet site which is trying to collate any information that we see out there on new papers or news stories or meetings of interest related to microbiology, the built environment. We would love it to get other people who work on microbiology, the built environment, to blog at this blog, or if you have a blog that you work on related to microbiology, the built environment, we can build a feed into our blog. I don't know how many people do this. Yesterday, I posted a survey to my blog and to Twitter about microbiology-related blogs, and some people were suggesting, oh, they're only about 10 or so of interest. And we've already got a list of about 65 microbiology-related blogs, many of which none of us were familiar with but seem very interesting. We have a list of about 20 built environment-related blogs, and there are many at the interface, and they're really a great way to just sort of build community and, again, keep up to date on things that are going on in a field that is rapidly changing. We're experimenting with other forms of social media. I personally think Twitter is an incredibly valuable system for keeping up with scientific information, whether it's at conferences or information about new papers or new developments. We don't have a lot going on in Twitter with this microbiology, the built environment project, but we're experimenting with ways to do that. And again, we're playing with basically any type of social media site to try and leverage it into a scientifically valuable communications venue. So one thing you can do with all of these that you may or may not be familiar with is, if anybody out there has a Twitter feed of interest or a blog of interest or some other thing that they post to, news stories, you can pull in that information to a central location where everybody can go and see all of the feeds from those particular sites, and then, you know, find out what's going on in that sort of entire arena through those feeds. And the last thing I want to mention related to this, which I think is the solution in the long run to getting a rapid acceleration in this area of microbiology of the built environment, is to teach everyone in this field how to participate in this type of sharing rather than doing it just via us. So we think that, for example, people should be posting presentations like slides from talks on social network sites like SlideShare, where people share slides and even audio. I'm recording the audio of my talk right now, even though ASM will probably get pissed off. And I will be posting later the slides and the audio of this talk. And that way you basically, rather than having a meeting of 100 people, if you want to share, this doesn't mean everybody has to share, but if you want to share, we can teach you how to share more broadly. If you have a new paper, we can help you get information about that paper out to a broader community. If you have a new data set, a new technique, interesting news story, again, we're not telling people that they should have a video camera attached to their head and record everything that they're doing, although, might not be the worst thing in the world. But if you have any interest in people finding out about something you've done, again, like a paper or a meeting report or whatever, we would love to help you learn how to share that information with others and broaden this community that is interested in microbiology, the built environment, such that it's not just the Sloan Foundation funding it, but it's every other government agency on the planet and private foundation, because it is an important, interesting area. So I think I'll just stop there. And thanks for listening to the substitute for Brendan. And if you have any questions, we have a minute or so, or a couple minutes. Thanks. Yeah, sure. Get up. If you can use the mic, that would be better.
I could probably just yell. My name is Brandon. I'm from the University of Arizona. I was wondering if you thought that the microbiome of the indoor water